Hello everybody, this is Dr. Aditi, your instructor, and we're going to be talking about epithelial tissue specifically today. So by the end of this video, you should be able to explain the structure and function of epithelial tissue and where it's located. Also list the key features of epithelial cells. Also be able to distinguish between the various varieties of cells. So let's get cracking. The first thing we want to talk about is an overall sort of purview of epithelial tissue. And we know that it is prominent in forming a barrier. I call it the barrier extraordinaire because it really is. It's not just about sort of safeguarding our body, but it also regulates what enters in and exits. So it's like border control almost. Also, it absorbs nutrients and can block harmful pathogens. So see, there's a lot more going on. And of course, a unique feature of this type of tissue that it has the regeneration ability, right? It's able to regenerate super fast. Like think about when you have a scratch or a wound and how quickly things heal and form, right? So this is definitely a unique quality of epithelial tissues. But the other thing I wanted to mention is that, we think of epithelia as forming boundaries, but often we may think of it just from the outside world. And it's not just the outside environment that it's creating the boundary for, it's actually doing it internally, right? Otherwise our organs would be a big hodgepodge in our body, right? So epithelial tissue also compartmentalizes a lot of our organs, enabling us to really monitor what comes in and what comes out of each organ. Due to their specific needs. So just a lot of aspects of epithelial tissue that we may not often think about, but their role is quite profound and vast. All right. Well, yes, there are different types of epithelial tissue, right? Obviously, and this probably just makes sense to you anyway. The epithelial tissue you have on your skin is going to look very different than what's in your glands versus what's in your GI tract, reproductive system, and blood, right? There's different epithelia there. And then, of course, your urinary system. And we're going to go through each of these areas and what specific type of tissue resides there. So as we unravel the key points on epithelial tissue, I thought I would show you a strategy that I use, which is the fill in the blank. And this is actually an exercise that I have in my ebook that accompanies each episode that I release. And this exercise, basically, you've got to figure out the key words and key points and fill them in the paragraph. And it's a really effective exercise if you're trying to learn anything and trying to get to that stage of active recall, where you're able to explain something without any notes or any prompts whatsoever. So it's a really effective learning strategy, and I thought I would use this opportunity to sort of show you how. So let's go through it together. First of all, epithelial tissue has many important functions. It provides, and you probably know this, protection in the skin, helps with absorption. It also helps with transportation and uses cilia to do that, aids in secretion and excretion, like in the kidney tubules, and gas exchange, like in the lungs or alveoli. Now, it allows for smooth gliding between surfaces as seen in the mesothelium. Now, this is something you may not think about, right? We always think about it forms a barrier, but it also lubricates so that there's no friction between our organs or other structures in the body. Epithelial cells continuously renew through mitosis, but don't have blood or lymphatic supply, and they rely on diffusion for nutrients. And unlike connective tissue, epithelial tissue or cells have no space between them, right? There's no extracellular fluid between them. The cohesion that's very strong with epithelia actually comes from special cell adhesions, molecules, and junctional complexes that are anchored in the basal lamina, which along with connective tissue forms like the basal membrane. But epithelial tissues also show structural and functional polarity, meaning that parts of epithelial cells have distinct roles. So there's this major polarity. So you can see that is an example of how you might use a fill in the blank example. But To summarize, epithelial line and cover our entire body except articular cartilage and the enamel of tooth, and like the anterior surface of the eye, or the iris, sorry. And then basic functions of epithelia we sort of went over, but just remember gliding between surfaces is also one of them. And then the other thing I wanted to mention again is a little bit more about the polarity. Epithelia have structural and functional polarity, like different parts have different and distinct roles. And we're actually going to explore that further as we move on. Okay, here we're going to talk about the 3 embryonic layers that epithelial tissues or cells derive from. So the lining of the skin and parts of the mouth and nose and the anus actually develop from the ectoderm, right? And then the digestive is your endoderm. And then the epithelia that lines our lymphatic or cardiovascular system derives from the mesoderm, and it's also called the endothelium. So ectoderm, the way I remember is ecto means on the very top, right? So that's going to be the parts of your mouth, nose, and anus. It's like the surface level. And then the parts that go in the digestive system, I remember that easily because endo has the word D for endo and D for digestion. And then lastly, the lymphatic and cardiovascular, I just remember that's going to be, it's in the middle of your body. So M for middle and M for mesoderm. So whatever works for you, but that's a strategy that works for me. Let's embark a little bit more in understanding the polarity of epithelial cells. So epithelial cells exhibit polarity, which basically speaks to the differences in structure and function between the apical part, or that surface facing the surface of the cell, and then the basal part, which is at the bottom, and that is close to the underlying body structures. So the basal lamina is at the bottom right there, and it's a mixture of glycoproteins and collagen, and that's sort of the attachment site for epithelium. So not only is that the attachment site, but what happens is this basal lamina is sort of the dividing part where it separates the epithelium from the underlying connective tissue. Now the basal lamina is going to be attached to something called the reticular lamina. And then that is actually secreted by connective tissue. And this forms the basement membrane, which holds everything together. So let me recap. We've got specialized epithelial cells, right? And then at the bottom, we've got basal lamina and reticular lamina. And that all forms the basal membrane. Okay, now epithelial cells are characterized by this polarization, and it's based on the distribution of organelles and membrane-bound proteins between the apical and basal surfaces. So some structures are going to be found only on the apical side, right? And others might be more on the basal side. So for example, cilia, which is what you see up here, right? That's going to be on the apical surface. And these cilia, we need to talk a bit more about them because they're very, very important extensions. There are these microscopic extensions of the apical cell membrane that are supported with these tiny microtubules. And what they do, they sort of beat in unison, and they move fluids as they trap particles, pollutants, dust, whatever. And you'll find these ciliated epithelium in various areas. Obviously, you know the trachea, but they're also present in the ventricles of the brain because it helps circulate the cerebrospinal fluid. And the ciliated epithelium basically, keeps things flowing in that area, but like in your airway, it also forms this what we call the mucociliary escalator because it sweeps these particles of dust and pathogens that are trapped in the secreted mucus, and it sort of pushes them towards your throat so that you can swallow it, and either it goes into your stomach or you cough it out. And often, I know in patients or people who smoke, the cilia are often damaged, and therefore often it's harder to get all that mucus out of the body, and hence you see people who smoke might cough a lot more, for example. But cilia have a very important function in epithelium, especially from a protective standpoint. Okay, so just to recap the key features, it's highly cellular, it is avascular, for instance, no blood vessels across the basement membrane to enter the tissue. All nutrients have to come in or out by diffusion or absorption of the underlying tissues or surface. So they really rely on the underlying tissues, right? And that's why the ability to connect to the underlying tissue is going to be very important. Also, we know that epithelial tissue are capable of rapid regeneration, replacing damaged cells and dead cells, and it enables that capacity of sloughing off damaged or dead cells as necessarily, especially in keratinized epithelia. Okay, we've talked about epithelia not being separated by intracellular material, but instead, there are these close connections that occur, right? And there are three basic types of connections that allow varying degrees of interaction between the cells. We've got tight junctions, anchoring junctions, and gap junctions. So let's start with the tight junction. And just like the name implies, this to me is like tight border control. This junction separates the cells into the apical and basal compartments. And when there are two adjacent epithelial cells in a tight junction, there's no extracellular space between them. So the movement of the substances through this extracellular space between the cells is actually blocked. So this enables the epithelia to act as very highly, tightly controlled selective barriers, hence the name tight junction. And you can see the location of these that are shown in the bottom corner here. Right? So this is going to be a tight junction. Now let's look at anchoring junction. This includes several types of cell junctions that actually help stabilize the epithelial tissue, right? Hence the name, anchoring. They're common on the lateral and the basal surfaces, which makes sense. Obviously, you want the anchoring at the bottom and you want it on the sides to keep things very stable and strong. But it also provides flexibility. Now, there are three types of anchoring junctions. And I don't know, when I look at this picture, it reminds me of aliens, because they're these like finger-like projections or whatever. But desmosomes are the top picture that you see there, and they occur on patches on the membranes of cells. And these patches have structural proteins on the inner surface of the cell's membrane. And there's an adhesion molecule very characteristic of desmosomes called cadherin. And it's embedded in these patches. And what it does, it has these finger-like projections that you see that look very alien-esque. And what it does, it comes out and links with other molecules, basically holding everything together. Now, hemidesmosomes, which you see at the bottom, are very similar to desmosomes, but essentially they look like half a desmosome, right? They've got these finger-like projections, but only on one side. And these link to extracellular matrix, for example, in the basal lamina. So, they're just going to need this projection on one side because the other side is connected to basal lamina. Now they're obviously very similar to the desmosomes, but one distinct difference, obviously you can see the difference being that it looks like half of a desmosome, but the other difference is that instead of cadherins, it has a different protein called integrins. So that's something you definitely want to remember. All right, then we've got adherence, and that's that carpet-looking like structure that you see on the top corner of the slide. Now, adherent junctions use either cadherents or integrants, so it can use either depending on whether they're linking to cells or matrix. So that's what you want to remember. When you're connecting to matrix, it's going to be integrants. When you're connecting to another cell, you're going to need cadherents. So just remember C for cadherents, C for cells. And these junctions are characterized by the presence of something else, though. They have a contractile protein, which is called actin, and that's going to be on the cytoplasmic surface of the cell membrane. And what the actin does, it connects with isolated patches, and it forms like this belt-like structure inside the cell. So, I mean, there's a lot more to it, but that's all you really need to know, that these junctions are key to how the epithelial tissue is going to look, how it's going to structurally unfold. And they do have some unique characteristics, each of these junctions. Okay, lastly, forgot about this, the gap junction. So in contrast to the tightness of the anchoring junctions and the tight junction, the gap junction is a little bit more loose, I would say, because it forms an intercellular passageway between the membranes of adjacent cells, and it does it to facilitate movement. So remember, tight, it restricts any sort of passing through. Anchoring is to just hunker down, and gap junctions is what facilitates movements of small molecules and ions between the cytoplasm and of the adjacent cells. So these junctions allow electrical and metabolic coupling of adjacent cells, and it can also coordinate function in sort of large group of cells, right? Because you need a lot of communication, and this was what allows it to happen or occur. All right, so now we're going to talk about epithelial tissue and their shape, right? They have very characteristic shapes unique to epithelia, and this to me is the most fun part of epithelial tissue. I had fun doing this, and I hope this is an activity that you might ponder and do yourself. Just grab some Play-Doh and see if you can actually model these cells, which is what I did. We've got the squamous, which looks like a flat pancake, cuboidal, which looks like a cuboid, and then columnar, which looks like a columnar, and this one's actually ciliated on the top. But basically, these are the three shapes of epithelial tissue. And as I said, if you get a chance, grab some Play-Doh and give it a go, because when you model it, you add this kinesthetic element to learning, which can be really helpful in retention and mastery. Okay, so as far as classification of epithelial tissue, I'm going to break it down, right? Whenever there's a name of simple, that means one layer. That's something you need to just memorize. Stratified means more than one layer, right? Could be two or it could be several. Pseudo-stratified means fake layer, because pseudo means fake. So this one looks like there are lots of layers, but in actuality, it's just one layer. And then lastly, we've got transitional, which basically means there's a medley of cells. There's lots of different cells involved, or shapes and sizes. Okay, so again, grab some Play-Doh and see if you can get these sort of modeled and figured out. It's just a fun activity, and it can really help you learn the epithelia. So let's start with simple squamous epithelium and stratified squamous epithelium, and let's take a peek at the differences. So that's simple squamous, and for this, simple means one layer, right? So we're going to have one layer of epithelial tissue, squamous being the flat pancake-like. Now this one is stratified squamous epithelium. So what are you going to do? You're going to take those squamous flat pancake-like cells, but you're going to make more than one layer. And unfortunately, I didn't have any more Play-Doh left, but I would have made more layers or rows if I could. Okay, let's move on to the other side, which is cuboidal cells. Simple cuboidal epithelium. So this is going to mean one row of cuboidal cells, right? But now the next one is stratified cuboidal epithelium. So we're going to take that row and we're going to add another layer because it's stratified, right? So do you see the difference? Right? Easy peasy. That's what you... Before I go into the classification of epithelial tissue, let me just preface it with this. It may seem awfully unnecessary and overwhelming because the names can be so vastly long, and it might be overwhelming to you. However, I promise you, once you figure out the breakdown of how to read the name and what it means, it is so simple. And once you know this strategy, this sort of breakdown, you're going to be able to figure out everything, almost everything you need to know about epithelial tissue just from the name. So let me break it down for you. The first part of the name is always going to be the arrangement. So it's either simple, stratified, or pseudo-stratified. The second part of the name is going to be the shape of the cell. Is it squamous? Is it cuboidal or columnar? The third part of the name is going to be something regarding a specialization or lack of a specialization. If there's no specialization, it's going to be blank. Then the last part is that you're going to add the word epithelium to it. And that's how you name epithelial cells. Let's give it a go. So here is a group of cells. We know the arrangement is going to be stratified, right? Because there's more than one layer. 2, what is the shape? It's going to be columnar because we can see that they're tall, long cells. Is there a specialization? No, there isn't. So you leave that part blank. And then 4th is you just add the word epithelium. And if you do that, then you should be able to come up with the name of the cell, which is stratified columnar epithelium. See how easy that was? Once you figure out the breakdown, it's really quite easy and so helpful because it gives you so much information about the cell type and where it's located. You can figure that out once you know this. Let's try our 4-step formula on this histology slide, right? So let's see. The first thing you'd want to know is how many layers. And we know it's just one layer, so that's going to be the simple arrangement. And then the second word is the shape. So what does the shape look like? And we can see that these are going to be cuboidal because they're sort of these box-like cells. Is there any specialization that we should be noting in the nomenclature? No, there isn't really. And then we just add the word epithelium. So this is going to be your simple cuboidal epithelium. And this is epithelia that is very active in secretion and absorption of molecules. So you're going to find them in the lining of the kidney tubules and in the ducts of glands. All right, let's give another histology slide a go with our 4-step naming process. So this is a slide I got from the library, and it was actually under simple columnar ciliated epithelium. And, you know, now this is probably a 200 power microscope slide. So I do find that it's easier to name the cells at a higher power, like 400 or so, because you can really see, especially when you're trying to distinguish between pseudo-stratified and simple, for example. So it is one layer, it's going to be simple in arrangement, and we know that these are columnar cells because we can see the shape. And then we want to know any specializations. Well, we know there's ciliated aspects to this, and then we're of course adding the word epithelium. So simple columnar ciliated epithelium is the answer here. Okay, let's have another go here with another histology slide. Now this is going to be a little different, right? This is your columnar epithelium, but it's pseudo-stratified because it looks like it's stratified, but instead of several layers, it's just a single layer of irregular shaped and differently sized columnar cells. So this is called pseudostratified epithelium. And the nuclei of these cells appear at different levels rather than clustered at the basal end. And that's why it gives this arrangement of stratification when there actually isn't any. So pseudostratified columnar epithelium, but we want to add ciliated because we see that there's some cilia in there. So pseudostratified columnar ciliated epithelium is found in the respiratory tract, where maybe not all of the cells have cilia, but some do. So again, this is an example how you could figure it out. So hopefully this was helpful and makes sense. Oh, I forgot. I've got one more slide in here to show the histology of transitional epithelium. So as you can see on the slide, the shapes aren't very defined, or they look like several different shapes, right? So this is called transitional epithelium because there are many shapes and there's no specialization here. And they're called this because there's this gradual change in the shapes of the apical cells as in the bladder. And a change occurs as the bladder fills with urine. Now, these cells are only found in the bladder, or the urinary system, I should say, more specifically, the ureter and the urinary bladder. So when the bladder is empty, this epithelium is going to look more convoluted and has more of cuboidal apical cell appearance. However, as the bladder fills with urine, this epithelium also stretches, and therefore the cells are going to look from being cuboidal to more squamous because they're sort of stretched out, right? So when the bladder is empty, it's going to look very thick and has this sort of multi-layered look, but then as it gets stretched out, it's going to have more of a less stratified look. And that's why it's called transitional, because the cells look like they're transitioning into different types of cells. And that's why we consider it a hodgepodge or medley of cells in transitional epithelium. And again, it's unique to the bladder and urinary system. Now that we know the classification of epithelial cells, what we need to do next is sort of look at the actual cells, their function and their location. And typically when you know one, you sort of know the other. But let's get started here on simple squamous epithelium. So this is going to look like thin scales, right? It's that single layer, almost like a sheet of paper that sort of covers everything. The nuclei are going to be flat, horizontal, and sort of elliptical in appearance. The endothelium is epithelial tissue that lines the vessels of the lymphatic, cardiovascular system, and basically it is made up of a single layer of these squamous cells. And simple squamous cells actually are very important because the thinness of the cell is what enables rapid passage of gas exchange or chemical compounds. Like obviously in the alveoli of lungs, it's going to be present for gas diffusion. And then the segments of the kidney tubules are going to have this too. And the linings of capillaries are also going to be made of epithelial. And then obviously lymphatic vessels, as you already know. So lastly, don't forget that the mesothelium is a simple squamous epithelium also that forms the surface layer of serous membranes. Basically lines the body cavities and internal organs so that it provides lubrication and enables organs to slide with each other without any sort of friction. Simple cuboidal epithelium are more like the nucleus of box-like cells that appear round and are generally located at the center of the cell. Now, these epithelia are active in secretion and absorption of molecules also, and so simple cuboidal epithelia are going to be observed in the lining of the kidney tubules or the ducts of glands, right? They're going to be very prominent in those areas because, again, they're for secretion and absorption. Okay, next we have simple columnar epithelium, and the nucleus of these tall column-like cells tends to be elongated, and they tend to be at the basal end of the cells. Like cuboidal, this is very active in absorption and secretion of molecules, which you're going to see in the digestive system, for example, parts of the female reproductive system also. Now, as you know, it can be ciliated or not, right? Ciliated columnar epithelium is composed of simple columnar epithelial cells with the cilia on top on their apical surfaces. We learned that earlier. And these are going to be found in the lining of the fallopian tubes and parts of the respiratory system, where basically their job is to transport, mucus and enzymes perhaps, or even pollutants basically in the organs. All right, now we've got pseudostratified columnar epithelium. Now this looks stratified, but it really is just a single layer of irregularly shaped and different size columnar cells. And basically, in pseudostratified epithelium, the nuclei of neighboring cells appear at different levels rather than clustered, and that's why it gives it that appearance. But we're going to see this in the bronchi, trachea, and much of the upper tract. Next, we have stratified squamous epithelium. This is going to be the most common type of stratified epithelium in the human body, and the top layer sometimes can be covered with keratin. So we're going to talk about that later, but examples of where this is going to be found is the lining of the esophagus, the mouth, and the vagina. And the specific quality of squamous epithelium or simple squamous epithelium, stratified squamous epithelium is quite a mouthful, is that, cells can slough off. We want them in abrasive areas to protect the body. And what happens is these cells are sloughed off, but they also regenerate super fast. So that is a very good quality of this type of cell. And again, it's found in the esophagus, mouth, and in the vagina. Okay, then we've got keratinized squamous epithelium. Now, this is very specialized type of epithelium because it has these dead cells with keratin on top. And what it does, it covers parts of like our oral cavity, the hard palate, and even the gums. Again, it protects against abrasions, adds extra protection, I guess you would say. So really important that you recognize which parts are going to be keratinized versus which that might not. Stratified cuboidal epithelium is going to be present in your sweat glands, salivary glands, and mammary glands. And this is also considered protective tissue. Okay, then we've got stratified columnar epithelium, and this is going to be present in the male and female urethra and the ducts of some glands. And this again secretes and protects. So epithelia does have a huge protective role, as you can see. Then we've got transitional epithelium, where it looks like a hodgepodge of cells, and this lines the bladder, urethra, and ureters, and allows for distension and relaxation of the bladder. And it is unique to this specific area singularly. Let's do a quick summary so you have a nice overview of what type of cells are found where. So a stratified squamous epithelia is going to be in your oral cavity, and then of course it's going to be on your skin, but you're more likely to have some keratinized stratified epithelia here, squamous epithelia. And then in the lungs, we know that simple squamous epithelia is going to occur there. The trachea is going to have more pseudostratified ciliated epithelia. And then kidneys, you've got the cuboidal, simple cuboidal epithelia. The small intestine is going to have a simple columnar, again more for secretion and absorption. And then the bladder is going to have your hodgepodge medley of cells, or at least that look like a hodgepodge or medley, transitional epithelia. Okay, so that's your little overview. All right, and that's all I have for you today. I always try to leave you with a mind map to give you sort of this overview of the topic, and to remember that anything you learn is not in isolation, right? There's a bigger fabric here, bigger picture that you need to always be cognizant of. So in this mind map, you can see, I've started off with the epithelial tissue and then move on to the shapes that it comes in, any sort of special categorizations and arrangement. And remember the 4-step process in naming epithelial tissue or dissecting what it looks like from the name. That's what is going to be really important for you. And when you are creating your own mind map, I encourage you to put all this information on there. So as you can see, I've got each type of epithelium listed. And then what you can do is draw out the cells. That's what I recommend you do. Even though if you're rubbish at drawing, just draw it, and even try to draw out where they're located. And you don't have to draw it, you could just write it too. But it's really good from a kinesthetic standpoint for you to learn and really master the concept. So hopefully this was helpful. Do leave me a comment on what you liked or didn't like about this video, because it makes me a better instructor and it provides you with better content. And I'm going to leave you with Ollie, who's going to give you a riddle on today's topic to give us somewhat of a summative assessment on what you learned today. See you later. Bye.